Video, Mr. Phillips, and today we're going to be looking at how the nervous impulse, the action potential, actually spreads across the nerve cell, across the axon. And you might be given a question that sort of links to this. It might sort of be asked another sort of application question: what's happening at these different stages of depolarization, repolarization, hyperpolarization, and the resting potential? And this is quite important to us because it's during depolarization that the sodium channels can open. During repolarization, the sodium channels close and the potassium channels open, and then during hyperpolarization, which is the refractory period, that is the point where no channels can open regardless of any change in charge. And that's going to be very important today when we look at how the actual impulse spreads across the nerve itself. So what do we need to know? Well, if we just have a look at this, you might just need to, if you're answering that question from the previous slide, you might just need to look at this sort of answer to get it into your head. So first of all, depolarization in one area triggers the next area along on the axon. So depolarization in one area triggers the next one. So for example, what we're saying is if the sodium ions rush in here because they've been opened, they then will move down here, and that triggers the next sodium gate to open because the charge changes, and so on and so on. But because of the refractory period, because of repolarization, any that come in here that go back that way have no effect on this sodium channel here. Its poles will stay closed. So let's have a look at that in a little bit more detail. So first thing you need to know there before we actually look at the what causes the movement is, I think this point is very important. So the stimuli, and whether something is a strong stimulus or a weak stimulus, as we said in previous videos, is not to do with the actual the amount of sodium ions, because they're always the same, that all or nothing if you cross the threshold. It's all about the actual frequency. So if you get a higher frequency of sodium ions rushing through and a higher frequency of depolarization, you will get a higher level of stimulus, and the brain or the muscle will detect and respond accordingly. If you get a sort of a weak stimulus, so you get much more less frequent, that causes your weak stimulus. So then, how does this work, and how does it actually spread along the nerve? Because we've talked about opening and closing sodium channels, but why would that cause a message to go down the nerves? Well, effectively, if we have a look here at number one, okay, so the first thing that happens is sodium ions diffuse through. So we've talked about this loads already in the previous videos. Sodium ions diffuse through, high to low concentration. Once your first sodium ion channel is opened, and if it's that's the Pacinian corpuscle, it's because of the pressures cause that to open because of deformation. Now, what will happen then is the sodium ions will then increase in that area, and they will naturally diffuse along the axon inside of the cell. And when they do, they get to they increase the charge from -60 up to potentially -50 millivolts again, and that then would cause this sodium channel, which below the one I'm writing here, to open as well because the charge has changed. That then triggers a load more sodiums to come in, and the same happens again. Those sodiums diffuse that way, it gets to -50, it causes this sodium ion channel to open as well, and then that continues down the axon. Okay, so effectively the sodium ion diffuses along the axon from a region of higher concentration to lower concentration, diffusion, and then the sodium gate, its next one, which is initially closed, opens because of the change in charge due to sodiums moving along, and that opens up again. So this is an example of positive feedback where one thing increases the next thing. However, if we go back to just the middle one, so if I just get rid of some of these arrows for a second, let's say that the sodium channel in the middle has now just opened, and obviously some of those ions will be going forward. Some of them, however, will be going backwards because diffusion, if they're high here and lower back there because they've already moved all the way down, some of them will move backwards, but it will not open this sodium channel again. This sodium channel will be closed, and the reason that this sodium channel is closed is because it's in the refractory period or the hyperpolarization period, where it doesn't matter for a few minutes what the charge is, it will remain closed as a result. And effectively this continues down the whole neuron. So what happens is you get this happening, the sodium ions rush in from the one area and they diffuse down and they cause that one to change charge, so that causes more sodium to come in, and so on and so on. Yes, they diffuse back, but when they diffuse back, it's always in hyperpolarization, or it's always in the refractory period, so it can't cause it to open. So the movement of sodium ions along the neuron increases the potential difference high enough to open the next sodium channel with via positive feedback. And as you can see here, it sort of shows almost that it looks like the charge or the action potential jumps from node to node. So if you remember, these gaps are called the nodes of Ranvier, and they jump and it jumps again and it jumps again. However, in real life, there's nothing to actually jump in. It's just that the sodium ions can't open any channels until we get to the edge of the myelin sheath, and that's where the channels open and the charge change. So it makes the charge look like it's jumping from node of Ranvier to node of Ranvier, and that is causing what we call saltatory conduction. So the myelin sheath is really important. Okay, so there are gaps in the myelin sheath. Said these are called the nodes of Ranvier, and the voltage-gated ion channels are only found here. So the action potential effectively jumps the short distance from node to node, and that process is called saltatory propagation or saltatory conduction. And this is why the myelin sheath is so important, because it increases the speed of node to node as you have bigger gaps because of the myelin sheath as a result. So myelinated axons conserve energy because there's less energy, sodium potassium pumps where the energy being used, and it effectively can go, if you don't have myelination, you have a maximum speed of about 1 meter per second, which is obviously pretty quick, but myelinated neurons can travel at 120 meters a second, so making the nerve impulse much more quicker. And again, if you were to compare the two between between myelinated and non-myelinated, myelinated nerves, the ones with Schwann cells and fat layers around the outside, they are much longer generally, and they have much faster conduction rates, whereas non-myelinated, which is represented here, these ones are much shorter generally, and they have much slower transition rates. Non-myelinated tend to make up the central nervous system, whereas other ones tend to make up the myelinates. So the action potential of a neuron is basically affected by 3 things. The temperature, the higher the temperature, the faster speed, so warm-blooded animals will have higher responses than cold-blooded ones. The axon diameter, so it's a bit like resistance in physics. The larger diameter, the more sodium ions are able to diffuse through it in one go, and therefore the faster speed. And some marine invertebrates that live at very low temperatures have developed thick axons to speed up the relative speed of their impulses. And the last one, the myelin sheath. Only vertebrates will have myelin sheath, and this speeds up the rate because you can do saltatory conduction, where it jumps from node of Ranvier to node of